The information in this module is accurate and complete to the best of our knowledge. All recommendations are made without guarantee on the part of the author or the sponsoring institutions. The author and the sponsoring institutions disclaim any liability in connection with the use of this information.
Welcome. In today's lesson, we will talk about surveys of genome variation. We will look at different methods of estimating genetic diversity. These methods use different parameters derived from the observed diversity. We'll look at the number of alleles, the number of segregating sites, the number of singletons, the observed homozygosity, and the mean number of pairwise differences. In an ideal neutrally evolving population, these different estimators of genetic diversity should have the same value. But in reality, they are seldom the same due to an interaction of evolutionary forces in a population. We'll start with looking at number of alleles and heterozygosity. All of this is a way of providing summary statistics that'll give you a general idea to summarize the amount of variation, and that does not encapsulate all the information presented in data, but allow comparisons between populations and loci. The simplest way to describe the amount of diversity is simply to count the number of alleles or haplotypes. However, this approach is highly dependent on sample sizes, which is usually a serious disadvantage. On the right, you see the frequency distribution maps of variation. Plotting these frequencies in different populations are very good visualization technique. The next type of descriptive statistics is number of segregating sites. Segregating sites are positions which show differences between related genes in a sequence alignment. The proportion of segregating sites within a gene is an important statistic in a population genetics, since it can be used to estimate mutation rates. To calculate the number of segregating sites, we could count the number of nucleotides that vary between the entire set of aligned sequences. In the picture, you can see segregating sites shown without connector between the two sequences, and the calculations. Another type of descriptive methods are looking at gene diversity and heterozygosity. Gene diversity at a locus is defined simply as, if you look at this formula, the number of alleles multiplied by their frequency. In simple case, for diploid loci, this statistics is referred to as measure of heterozygosity, which becomes virtual heterozygosity at haploid loci. This measure of age, or gene diversity, can be corrected for sample size by multiplied by n divided by n-1, where n is the sample size, to give an unbiased estimator of population heterozygosity. The next descriptive method is nucleotide diversity of pi. Nucleotide diversity describes the probability that two copies of the same nucleotide are drawn at random from a set of sequences will be different from each other. The advantage is that the expected value of nucleotide diversity is independent of sample size. Nucleotide diversity is denoted as pi, and it looks at the frequencies of different alleles and proportion of different nucleotides between them. Once you calculate nucleotide diversity, you could build the next descriptive, which is mismatch distribution. The mismatch distribution is a histogram obtained by counting the number of pairwise comparisons that share the same number of differences between the two sequences. You can construct a distant matrix by counting frequencies of pairwise differences between a set of 5 sequences, like in this example. The shape of mismatch distribution depends on the history of a population. The mismatch distribution is a good example of a descriptive summary statistics that overlaps with inferential methods. For example, smooth bell-shaped distribution indicates a period of rapid population growth from a single haplotype, while a ragged multimodal distribution indicates a different situation. For example, when a population whose size has been constant for a long time. So when we look at all these descriptors of genetic diversity, what have we learned about the species? Well, let's look at our own species, the humans. Humans, despite their worldwide distribution, show very low amounts of variation compared to other species. Most of our diversity is still in Africa. This reflects our recent expansion from Africa, starting from a much smaller and more geographically restricted population. I prepared some review questions for you. What is the simplest way to describe the amount of diversity in the population? What is heterozygosity? What is the difference between expected and observed heterozygosity? Can you define nucleotide diversity? Describe and interpret different shapes of mismatch distributions. How diverse are humans compared to the other organisms? What is the general pattern of human genome diversity around the world? This concludes today's lesson, Survey of Genome Variation. Next, we will be studying the topics of individual differences.